Good afternoon. Welcome to today's webinar, Understanding and Overcoming Antibiotic Resistance in the GI Tract. Today's presenter is Dr. Amy Rolfsen. A little bit about Dr. Amy. She is a naturopathic doctor and a medical consultant. Her areas of clinical interest and expertise are women's health, children's health, immunology, and gastrointestinal health. Dr. Rolfsen is continually working on mastering her craft. She runs her own clinic where she sees patients in person and also consults with international clients. She works with people who are seeking guidance in understanding their health and taking steps toward perfecting their physical, mental, and emotional health. Dr. Amy is also part of our medical education team and does consultants for DSL. And we're just very pleased to have her with us today. And she's bringing us a very interesting topic. And so, Dr. Amy, we're glad that you're here, and we look forward to hearing what you have to say. And so with that, I will turn it over to you.
And thank you, Jeff, for the wonderful introduction. And thank you, everyone, for joining us this afternoon. We are here today to discuss antibiotic resistance as it pertains to the gastrointestinal tract. And during the presentation, I'll be moving fairly quickly through a number of topics. First, I'm going to start by outlining the basic mechanisms of antibiotic resistance, also touching on how biofilms can contribute to resistance. Then I'll go on to review the antibiotic resistance markers on the GI map, as well as how to pick up on possible resistant organisms on the test. Finally, I'm going to go on to talk about some strategies for overcoming resistance using botanicals in combination therapy. So let's get started. Antibiotic resistance is when bacteria develop the ability to survive the antibiotics that they were meant to be susceptible to. Antibiotics are a fantastic and life-saving invention. With the advent of antibiotics, we were able to treat conditions that were previously untreatable and perform routine surgery without the risk of life-threatening infection. Ever since antibiotics were introduced, however, bacteria have been developing methods to outsmart them. When antibiotics are used, organisms that are susceptible to the antibiotic will be killed, while organisms that are resistant will survive and go on to form colonies. Since beneficial flora can also be negatively affected by antibiotics, we will get two effects. First, we will be selecting for resistant strains of the organism you are trying to eradicate, and second, we will be reducing numbers of protective organisms. The Center for Disease Control recently published its 2019 risk report with some interesting figures. Since the last threat report in 2013, the rate of infections from antibiotic-resistant strains actually decreased by about 18%. The numbers are still incredibly high. In the US each year, there are roughly 3 million infections associated with resistant strains, resulting in 45,000 deaths. I've included the stats for C. diff here in these calculations. So even though numbers have gone down, new organisms have been entered into the threat list. And here's the current threat list. It's broken down into urgent, serious, concerning threats, and a watch list. So as I said previously, the number of people affected by resistant infections has decreased in the past few years, which is wonderful. But new resistant organisms have become threats. And before you start taking screenshots, know that you will be receiving a resource with all the relevant information on these slides. And more from the CDC threat report. Here's a quote from the director of the CDC, Dr. Robert Redfield, urging us to stop relying only on new antibiotics. New antibiotics are in the works, and according to the CDC, there are 42 new antibiotics in development. Only about a quarter of these have a new mechanism of action, and about 11 of these antibiotics are being developed to target current threats. Development of these new drugs is a long and drawn-out process, going through 5 different steps of development and approval. So it could take years before even one of these new drugs makes it to market. And with the rapid evolution of bacteria, there's no telling how long it will be before resistance develops. So really, this is a call to all of us to come up with different methods for handling infections. And the report also serves as a call to action against resistance. So part of their suggested solution is choosing a method of testing for resistance before giving an antibiotic. Here at Diagnostic Solutions Lab, we do testing for resistance genes as part of our GI map. I'll go over this testing in detail in a few moments. There are also a number of potential tools listed to combat resistance, including vaccines, antibodies, bacteriophages, and fecal microbiota transplant. And while it wasn't mentioned in the report, today I'm going to suggest another tool that we can use to target resistant strains, and that is botanical medicine. And I will explain exactly what I mean by this a little bit later in the presentation. So why are the bacteria winning in the battle of antibiotic resistance? Bacteria are able to easily and rapidly share genetic information. So plasmids are a type of mobile genetic element that can transfer DNA between bacterial cells. Bacteriophages are a subset of viruses that can infect bacteria, and they can also transport genetic information between infected bacterial cells. Mutations in bacterial DNA can also infer resistance. Mutations can either add function, such as a protein that will block a binding site, or they can take a function away, like inactivating a gene that controls the expression of resistance elements. Induction is the turning on and off of gene expression based on conditions. Bacteria are very thrifty with their energy. They won't express any genes that aren't necessary for their current state. Under less than optimal conditions, however, bacteria can express dormant resistant genes. So in a normal growth medium, a bacterium might not be producing enzymes to break down antibiotics or efflux pumps to remove antibiotics from its cell. But once conditions change, like in the presence of even low levels of antibiotics or sanitizers, these genes can get induced and the bacteria can express their resistance. This is part of the reason why our systemic overuse of antibiotics, both in the medical field and in the food supply, can be so problematic. Antibiotics don't break down readily, so they end up in our water supply and in wastewater treatment plants at low levels, meaning that bacteria are floating around in a persistently low level of antibiotics. This can lead to persistent resistant traits in the dominant strains of bacteria. Bacterial populations can grow very rapidly. Some bacteria, such as E. coli, can double every 20 minutes. So if most of a population of bacteria is susceptible to an antibiotic, but a few members are resistant, we will be selecting for the resistant bacteria. And what starts out as even a small number of resistant bacteria can turn into a large colony in a short amount of time. They can then go on to potentially share their genetic information with other organisms. Bacteria have methods of signaling and sensing when other bacteria are around. Quorum sensing is when a critical population level of an organism is reached. At this point, the organism or organisms will express certain gene traits, such as those for virulence, competition, or biofilm formation. Get ready to hear this term again soon because I'm going to talk about quorum sensing a lot today. It's fairly clinically relevant. Moving on, the major difference between gram-positive and gram-negative bacteria is that gram-negative organisms have two cell membranes, while gram-positive organisms have only one cell membrane. Gram-negative organisms, because they have two cell membranes, have a special holding area between the membranes called the periplasmic space, right here. This allows the bacteria to deal with any foreign compounds within the periplasmic space without exposing the inner cell. Because of this, gram-negative organisms tend to have more successful resistance mechanisms than do gram-positive organisms. Gram-positives do still develop antibiotic resistance because once antibiotics pass through their cell membrane, they will be inside the cell. The resistance mechanisms of gram-positive bacteria have to be much faster and be induced much quicker than their gram-negative counterparts. There are a number of different strategies bacteria have of avoiding the effects of antibiotics. Many of these strategies are intrinsic, meaning they are part of the natural genome. I spoke earlier about a few ways bacteria can acquire new resistance. And I'm going to go over these, not because I want to bore you senseless, but because knowing the methods bacteria use to resist treatment can help us select our treatments very consciously. These are going to be your therapeutic targets for treatment of resistant organisms. So the first line of defense for a bacterium is to prevent access of the antibiotic to the cell. It will do this in two different ways, by altering uptake of the antibiotics and by pumping them back out again. So bacteria have varying levels of membrane permeability, which can be changed depending on conditions. Under normal conditions, there will be channels called porins that allow entry into the cell. There's one right here. If the membrane becomes less permeable due to a mutation or under expression of porin channels, less antibiotic can reach the inside of the cell. Efflux pumps are pumps that cross the cell membrane. There are a number of different types of efflux pump that have affinities to different compounds. Their major job is to recognize outside compounds and pump them outwards. And there's an efflux pump right there. Back into the interstitial space as a means of keeping the internal concentration of the compound down. This protects the cells from the antibiotics or other compound. For most organisms, efflux pumps are part of their natural genome. The expression of efflux pumps is normally very low, so while the genes may be present, efflux pumps are not being expressed and so will not be active. Their expression can be induced by the presence of antibiotics and other chemical compounds. They can also be induced in the presence of bile salts and fatty acids, indicating that efflux pumps may be expressed under the normal conditions in the GI tract. So the expression of efflux pumps will likely be different outside versus inside the body. Another really interesting aspect of efflux pumps is that they are not exclusive to pumping out foreign compounds. They are also used to disperse signals from inside the cell, such as those used in quorum sensing. So what that means from a clinical standpoint is that any agent you use to inhibit efflux pumps also has the potential to inhibit quorum sensing. Inhibition of quorum sensing can affect a number of factors, including expression of virulence and biofilm formation. So once an antibiotic reaches the inside of the cell membrane and begins to increase in concentration, bacteria have two more mechanisms: antibiotic modification and target modification. So bacteria have the capability, both inherent and induced, to produce enzymes to break down antibiotics. The most common enzymes found fall under the class of beta-lactamases that break down the beta-lactam antibiotics. And each antibiotic has a crucial target within the bacterium. For some, it is the cell membrane. For others, the target is the genetic material, protein synthesis, or cell division. Mutations can arise within a bacterium that alter the antibiotic target, changing its susceptibility to antibiotics. Biofilms technically fall under the category of antibiotic tolerance, not resistance. But biofilms contribute so much clinically that I thought I'd touch on them a little bit in today's presentation. For those who are unfamiliar with the concept, a biofilm is a substance secreted by the bacteria themselves. It's made of polysaccharides, proteins, DNA, and other particles. It surrounds and protects the bacteria and often allows the colony to adhere to a surface. Often within a biofilm, you will have numerous types of organisms living together, and this gives them the perfect chance for sharing genes as well. But biofilms are not totally bad. They can play an important role in normal and protective GI microbial populations. For example, lactobacillus can be a biofilm producer. Many organisms have the genetic potential to produce biofilms, but they will only be expressed in the right conditions as a distinct growth phase. Biofilm formation can be induced by many things, including low levels of antibiotics or quorum sensing. For an organism to form biofilms, it must first attach to a surface. There are a few natural substances I'll mention later that can affect both bacterial adhesion and quorum sensing. I'll also be discussing a few of the many options for disrupting existing biofilms. So biofilms can restrict antibiotics in two ways from entering the cell, entering into the matrix. But the first is that they can actually block the antibiotic, and the second, they can absorb the antibiotics into the biofilm matrix. So not actually blocking the entry, but just sucking it in. And the GI map tests for quite a few organisms with biofilm producing potential. I've listed all of them in the resources you'll be receiving, but the ones that really stand out to me when I'm interpreting a GI map test are E. coli, H. pylori, Pseudomonas, Klebsiella, Proteus, and Candida. So now that we've established the mechanisms of resistance, we can make a list of possible therapeutic targets. Here we've got efflux pumps, quorum sensing, bacterial adhesion, cell membrane permeability, bacterial DNA, enzymes, and biofilms. I'll be coming back to this list later in the presentation when we'll go over some botanicals to target each mechanism. So now let's go over page 5 of the GI map where we'll find the antibiotic resistance markers. So testing for antibiotic resistance can really help target your treatment. It can improve accuracy of your diagnosis, it can help you choose the right antibiotics, and it can also help you avoid the wrong antibiotics, which is just as valuable as choosing the right antibiotics. It can also help to identify the spread of resistant bacteria and the genes, whether they're being expressed or not. On the GI map, we're testing a large number of known resistance genes and reporting them on page 5. The test comes standard with 13 genes for H. pylori resistance, testing for antibiotics that are commonly used in H. pylori treatment regimens. We're also testing for genes that can be found in the universal microbiota, and I'll explain what that means later on. The genes we've selected are presented here because they are clinically relevant to common antibiotic treatments and because they've got the best volume of research behind them. And the genes you'll see on page 5 are dynamic, which means that they change as the microbiome changes. So they are not part of your genome, they are part of the organisms living within you. So within a few weeks or months, you could see these markers changing. If you're not familiar with ordering the universal microbiota resistance genes, here's a look at the GI map requisition form. If you check this box here, you will receive the universal microbiota resistance genes as well as the H. pylori resistance genes. The H. pylori genes come standard with the test, and the universal genes have to be added on. And this is a no-cost add-on to the test. So let's take a look at the H. pylori resistance genes. Each letter number code here represents a different gene. If H. pylori isn't detected, you'll see a little NA next to each gene, meaning that it's not applicable because we don't have H. pylori present. Here we're looking at the antibiotics commonly used in first-line therapy and second-line therapy against H. pylori. First, we're looking at the 2 antibiotics used in classic triple therapy, which basically triple and quad therapy share first-line therapy status depending on your region or resistance. So first, we are looking at amoxicillin and clarithromycin, which together with a proton pump inhibitor make up classic triple therapy. Resistance to amoxicillin for H. pylori is fairly low, but resistance to clarithromycin can be up to 30%. Fluoroquinolones are usually reserved for when triple and quad therapy are ineffective. Many practitioners don't use fluoroquinolones in practice, but knowing the resistance can still be helpful. And the resistance to fluoroquinolones is estimated at up to 22%. So down here we're testing for resistance to tetracycline, which is part of quad therapy. And quad therapy is usually tetracycline, metronidazole, a proton pump inhibitor, and bismuth. If H. pylori is detected, this is what the H. pylori resistance markers will look like. And here we can see that H. pylori is present, but there is no resistance present. So you'll see the word absent next to each of the genes here. If a resistance gene is present, it will be flagged red. So here we can see that this patient has H. pylori present with some resistance genes. One gene is present that codes for resistance to clarithromycin, making resistance positive to clarithromycin. There's also one gene present that codes for resistance to tetracycline. And so there is also positive resistance to tetracycline. Since clarithromycin is part of triple therapy and tetracycline is part of quadruple therapy, neither would be a great first-line option for this patient. And here we're looking at the universal microbiota resistance genes. Again, this is an optional no-cost add-on. You need to check the box on the requisition form to receive this data. These are genes that can be easily transferred between organisms, so they're presented as universal genes instead of listing individual organisms. The first section here codes for beta-lactamases, the enzymes that can break down beta-lactam antibiotics. Resistance in this section is quite common, and I want to point out two markers in particular under the beta-lactamase section, OXA30 and NDM1. These two genes code for carbapinamases, enzymes that inactivate carbapinams, which are often used as a last resort IV drug against multi or extensive drug-resistant organisms. So if you see these markers pop up on your test, you might be dealing with an especially resistant organism, or at least the genetic potential that could be expressed as resistance. The next section contains genes that code for resistance to the fluoroquinolones. Both of these genes code for a protein that blocks the antibiotic from binding to its target. Resistance to macrolides is fairly common. ERM A, B, and C code for alteration of the antibiotic target. These genes are normally dormant, but they can be induced under certain conditions. The MEFE gene codes for an efflux pump with macrolide specificity. The last section contains genes that code for resistance to vancomycin. Then A1, A2, B, and C all code for proteins that block antibiotic binding to target protein in the cell. To illustrate how dynamic these markers are, I'll show you a very brief case study of a woman who came into my office. She has a history of mycotoxins, chronic viral infection, chronic antibiotic use, and severe fluoroquinolone reactions. Here on her first GI map, you can see that her universal microbiota showed a lot of resistance genes. Her H. pylori was negative on this test, so I haven't presented the H. pylori genes. But you can see that she's got 3 genes for the beta-lactamases, 2 against fluoroquinolones. Just moved. Anyway, we are on to another slide. And here she is 4 months later, after some gut work, showing a very different picture. She is the same person with the same genome, but her microbiome is very different. The organisms that carried resistance genes are no longer present. This isn't a matter of gene induction or expression. What we're measuring is the genetic potential for antibiotic resistance. Quantitative PCR is especially helpful in that it can identify strains that are growing unchecked. The GI map doesn't just measure whether an organism is present or absent, it can tell you the extent of the overgrowth. If a patient has a history of recurrent infections from a single organism, we should always be considering resistance and tolerance factors. If a patient has a recent history of antibiotic use and is still showing large colonies of some organisms, that is also very significant. And here's another case study. Here we see a 47-year-old woman with chronic GI concerns. Within the past 2 months, she had been on a round of antibiotics. And when I look here at the Opportunistic bacteria page, I like to scroll down and look at the orders of magnitude in the result and compare them to the reference range to see if anything really stands out. And as we scroll down, we can see that yes, something here is really standing out. Look at the numbers of Pseudomonas there, which are growing at 8 orders of magnitude above the reference range. And also the numbers of Morganella are quite significant there at 3 orders of magnitude above the reference range. Based on the nature of Prevotella copri, I would say that the Prevotella here could be quite significant as well. And so what is it about that Pseudomonas that is allowing it to grow unchecked in such a large colony? What we do know about Pseudomonas is that it has a huge genome that codes for a lot of potential resistance mechanisms. We also know that it has the potential to create biofilms. So in this patient, I would be ensuring that my treatment was very specific for Pseudomonas and that I included resistance modifiers as well as biofilm disruptors. And here's another test we can use as a case study to illustrate how we might think through the resistance gene markers in choosing antibiotics. So this is the test of a 36-year-old woman who also recently had a course of antibiotics. Going through the test, she was negative for pathogens, and here she is positive for H. pylori. And there are a number of anomalies in the opportunistic flora section. Looking at the exponents here, comparing the reference range to the results, we can see that Klebsiella really is sticking out a little bit. Klebsiella, the genus, is growing at 3 orders of magnitude above the reference range, whereas Klebsiella pneumoniae is growing at 2 orders of magnitude above the reference range. Klebsiella is an organism that is reported to have resistance to a number of antibiotics. It also has the potential to produce biofilms and histamines. I'm going to leave out the intestinal health markers, not because they're insignificant, but because they're not what I'm talking about today. So let's move on to the resistance markers. So let's say hypothetically, we were planning to choose antibiotics to target the H. pylori as our primary target, but we would also like to eradicate the Klebsiella. Looking at the resistance markers for H. pylori, we can see here that there are no genes present that code for antibiotic resistance. This means that classic triple therapy or quad therapy would be options. Fluoroquinolones would also be an option. Okay, and moving on to the universal genes, we see some resistance here. We can see here that there is one gene coding for resistance to the beta-lactams, and there is one gene that is, two genes coding for resistance to the macrolides, ERMB, MEFE. And so if we think back to classic triple and quad therapy, amoxicillin is a beta-lactam antibiotic, clarithromycin is a macrolide, and so that would mean that triple therapy, which contains amoxicillin, clarithromycin, and a proton pump inhibitor, would not be a great first-line choice. You would be much better off using quadruple therapy if you were to choose an antibiotic regimen. And in a few moments, I'm going to talk about things we can do to augment treatments to make them more effective and to broaden our spectrum of antimicrobial action to also target Klebsiella, because quadruple therapy is not necessarily going to be effective against Klebsiella. So now that we've gone over the antibiotic resistance and the testing resistant genes, let's switch gears and talk about what we can do about it. First, let's clarify what our goals are in treating resistant organisms. We want them gone, but is it entirely our job to eradicate them? I don't think so. I believe our job is to bring the organisms down to a certain level until they are in equilibrium with both the normal flora and the gut immune system, and also making sure that there is not a significant amount of membrane permeability so that the gut is intact. And in other words, that means to regain gut homeostasis. So here are the basic strategies I'm going to suggest for overcoming antibiotic resistance. Antibiotic stewardship, the use of botanicals as antimicrobials to replace the antibiotics, the use of botanicals to augment treatments with either antibiotics or in combination with other botanicals. Finally, respect and support the immune system and gut terrain. So let's talk about antibiotic stewardship, or the proper use of antibiotics. And I'll be speaking about antibiotics in the context of a clinical setting, so not necessarily talking about the food supply. So antibiotics should be reserved for serious, life-threatening conditions. Mild and chronic infections can be treated effectively by other means, such as botanicals. When antibiotics are needed, test for resistance and choose antibiotics appropriately with as narrow a spectrum as possible. Encourage the proper use in your patients, and then do what you can to improve the efficacy of your antibiotics. There are some botanicals with good research showing that they can decrease the minimum inhibitory concentration of antibiotics, meaning that you need less antibiotics to get the same effect. As an alternative to antibiotics, you could choose to use botanicals to treat infections. Let's briefly go over some of my favorite herbs to use in practice to kill bacteria. First, I'd like to mention the research available in botanical medicine. So most of the data available are taken from in vitro studies. There are some in vivo data from animal studies and a very small amount of human data. To make up for this, we also have hundreds of years of history of safe and effective traditional use, and oral history is very strong in botanical medicine. So I'll be presenting the information here, which can give you some ideas for potential for therapeutic intervention, and whether you choose to use herbs in this way in your practice will be your clinical decision. In this presentation, I'm focusing my efforts on the treatment of resistant bacteria in the GI tract, and the difference clinically is absorption. The herbs I'll be talking about have great local action. If you're treating a systemic infection, you would choose different herbs and augment treatment to enhance absorption. So here's a list of herbs I use when treating GI infections. So I'm going to briefly go over a few of my standby herbs to use as antimicrobials. These herbs are well documented to have antimicrobial action, and I've used them in the GI tract with a great deal of success. This is not even close to an exhaustive list of herbs with antimicrobial potential, just the ones that are readily available and work well for me in practice. What the research suggests over and over and over again is that the use of the whole herb is superior to the use of standardized constituents. So while many practitioners are using extracts like berberine or artemisinin, therapeutic effect is likely to be greater when you use a whole herb extract, at least when it comes to resistant organisms. This is because herbs have hundreds of constituents working all together. And there is absolutely a place for standardized extracts in therapy, but if I had a choice of a standardized berberine or Oregon grape for treating a GI infection, I would take the Oregon grape every time. So berberine-containing plants, your Mahonia, Berberis, Hydrastis, and Coptis, to name a few, have been used historically as broad-spectrum antimicrobials. These are a mainstay in botanical antimicrobial treatment. In a few moments, I'll discuss their use as potential synergists as well. Garlic has a broad range of antimicrobial activity. It can be used for both gram-negative and positive bacteria, as well as fungal, viral, and parasitic infection. One of the great things about garlic is that it has been shown in vitro to be antibacterial against pathogens while sparing normal flora, such as lactobacilli. The best way to use garlic is either fresh or freeze-dried. Artemisia has a long history of use in Europe and Asia as an antimicrobial. It is effective against bacteria and parasites, and again is best used as whole herb extract. Oregano is a great antimicrobial that has broad spectrum action against bacteria, fungi, and archaea. And if you get a strong formula of oregano, you can do great things with it. I have some patients that take it down traveling in South America with them for if they get some gastroenteritis along the way, they will take a few oregano and feel better. Usnea, or old man's beard, is a lichen with really strong gram-positive antibacterial and antiviral action. If you look at the research on botanical antimicrobials, you'll notice something. The studies come from all over the world. India, Korea, China, Jordan, Tunisia, Turkey, and Pakistan. And there are all these great studies that they're investigating traditionally used herbs in a clinical setting. And what does this tell us? Does it tell us that we should travel the world to find the best antimicrobials? I mean, maybe. To me, this means that no matter where you are in the world, if you go outside, you can find botanical medicine to help you with what you need. It also tells me that there's a lot more room for research of native North American botanicals. And now it's time to talk about synergy. Synergy is the concept of the whole being greater than the sum of its parts. These botanicals can be used alongside antibiotics or antimicrobial herbs to potentiate their action. Some of the botanicals listed have antimicrobial activity on their own, but also contain constituents that perform other actions. Some botanicals have no antimicrobial activity, but can strongly augment antibiotic or antimicrobial therapy. So remember our list of therapeutic targets? Here it is reflected as categories of botanical synergists. You'll see that there are some herbs that fall under more than one category. I'll go through this target by target. After that, I'll mention a few of my favorite herbs to use as synergists in practice. Please remember that you will be receiving all these lists at some point after the presentation, as well as a chart with all the herbal actions. Take a look at the chart. On it, I've included the antibiotics that were used in combination with herbs in the research. For a few botanicals, the reduction in MIC, or the amount of antibiotic needed, decreased dramatically when combined with herbs. And I mean dramatically. For some botanicals, the MIC decreased by more than 200 times. And also know that this list is far from complete. I've limited my list to botanicals that can be sourced here in North America. The research is also far from complete, so I've included everything I was able to find. And so what would you do with these lists? First, look up the organism you want to get rid of. Find out what its potential mechanisms of resistance are. Also choose an antibacterial agent based on resistance testing, research, and clinical experience. Then address any possible resistance mechanisms with the botanicals on these lists. In some cases, one botanical will cover many bases. In other cases, you'll want to use a combination. From my experience with herbs, they work really well in combination. So I'm usually choosing 3 or more to work with. Some botanicals might not be appropriate in every situation, and once you get the hang of herbal energetics, you might find that some herbs match your patient's constitution better than others. But that's a whole different topic. So let's get started. Inhibiting efflux pumps can allow antibiotic levels to build up within bacterial cells. Most bacteria have efflux pumps as part of their normal genome that are either not expressed or they can be induced in the right conditions. So these herbs have been shown in vitro to be strong efflux pump inhibitors. You'll see that Artemisia and Berberis are on this list as well as on the antimicrobial list. These herbs have a lot of evidence of their activity against efflux pumps. Since plants are constantly exposed to microbes in the wild, they have great inherent ability to overcome resistance. Built-in efflux pump inhibitors are just part of the natural defense mechanism of plants. The same goes for the other targets we'll be talking about. Quorum sensing inhibitors will stop bacteria from sending out and receiving signals that would turn on virulence factors and biofilm formation. This is a list of botanicals and nutrients that have in vitro evidence of quorum sensing inhibition. As I mentioned previously, any agent that has the ability to inhibit efflux pumps can also inhibit quorum sensing. And any agent that can inhibit quorum sensing has the potential to inhibit biofilm formation. Note on this list that there are some common foods, like kale and berries. Also, I'm not sure if you've noticed by this point or not, but green tea is a huge player in overcoming resistance. Adhesion inhibitors will disrupt the ability of bacteria to stick to surfaces. Adhesion is the first step in colony formation, so if this function is taken away, new colonies will not be able to take hold. However, these agents will not necessarily be able to disrupt existing colonies, at least not based on their adhesion inhibition. Cell membrane disruptors can help antibiotics to gain entry to the cell. Here are a few herbs that have action against the cell membrane. These have the potential to augment any antibiotic treatment. Nootka cypress in particular is noteworthy here. It was documented in one study to potentiate the action of methicillin by 256 times against methicillin-resistant Staph aureus. Cypress also has activity against efflux pumps, so it is likely a multifactorial effect at play here. Enzymatic inactivation plays a huge role in antibiotic resistance. Here are some botanicals that have been shown in vitro to have action against bacterial enzymes. And then here is a long but still extremely limited list of biofilm disruptors. The botanicals here have the potential to disrupt early biofilms, which can be really clinically useful. In cases where you suspect long-standing biofilms, such as chronic infection, you may need to bring in stronger agents. If the organism is a known biofilm producer, it is a very reasonable strategy to include a few botanicals or agents from this list. If the infection doesn't clear as expected, you might be looking at more substantial biofilms and may need to choose something a bit stronger. So now it's time to go over a few of my special favorites. Since we need to keep regular stock in our dispensary, it makes sense to choose your favorites, and yours might be different from my favorites. So licorice has evidence for moderate antibacterial action and is commonly used as a strong antiviral agent. What's remarkable about licorice is its strong efflux pump inhibiting effects. So licorice will often be one of my first choices when efflux pumps are present. And I can hear a question forming. Can I use DGL? Yes, you could actually use DGL. So the G in DGL is deglycerized licorice, which means that the glycyrrhizin is removed. Glycyrrhizin is active against bacteria, but it's actually the glycyrrhetinic acid that is acting against the efflux pumps. And so you may lose some of the function if you use a DGL, but if DGL is a better choice for your particular patient, then choose the DGL. If you have the option, you may do better with licorice tea, tincture, or a solid extract. And licorice isn't for everybody. It isn't safe in pregnancy, it can raise blood pressure, and should only be used short term. Green tea is a powerhouse when it comes to overcoming resistance. Did you notice that it was on 5 out of the 6 lists I provided above? A great review paper by Dr. Eric Yarnell and Kathy Abascal did the calculation on dosage and estimated that the therapeutic benefits of green tea could be achieved at around 1 standard cup of tea daily. And I hear what you're thinking again. Can I use EGCG? Yes, you can. But you'd be missing out on the action of a few other and potentially way stronger constituents. Epicatechin gallate, ECG, and catechin gallate, CG, have been shown to reduce the amount of antibiotics needed to kill methicillin-resistant Staph aureus, and they actually reduced the MIC of oxacillin from, it tested positive at both 256 and 512 milligrams per milliliter, down to 1 to 4 milligrams per milliliter. Yet another example of how the whole herb can be more impactful than the standardized constituent. In one study on mice, green tea extract was used with levofloxacin, a fluoroquinolone, against enterohemorrhagic E. coli. The green tea and antibiotic group had greater survival rate compared to the antibiotic group, and on top of that had reduced organ damage, which often comes as a result of fluoroquinolones. So green tea can also be liver protective. Berberine-containing plants, again, show great synergy with a wide array of antibiotics. It was shown in one study to increase susceptibility of 3 organisms, Pseudomonas, Staph aureus, and E. coli, to ciprofloxacin by 33 times. It's worth noting that there is some evidence that berberine plants don't combine well with tetracycline. And there is also some documented resistance against the standardized berberine extract. So berberine-containing plants work best as a whole herb, and you can get the full synergy by using the root and the leaf together. Chinese skullcap contains a few constituents that can be tremendously useful, both as antimicrobials and as synergists. It can potentiate beta-lactams, fluoroquinolones, gentamicin, and tetracycline. It can also modulate the immune system in the right situation. Chinese skullcap is related to, but not interchangeable with, the commonly used nervine skullcap, so Scutellaria lateriflora. And we've already looked at garlic as an antimicrobial. It can also be used to inhibit quorum sensing, beta-lactamases, and DNA synthase. Garlic is best used either fresh or freeze-dried, and the same review paper I mentioned earlier by Dr. Yarnell and Abascal estimated the dose of garlic at 1 raw clove daily. So moving on, we discussed earlier the importance of the immune system in managing resistant and chronic infections. Gut immunology is extremely complex, and everyone will have different needs. So I won't go into it deeply today. I've got a few lists here, and there are a few agents I use on a regular basis in practice to modulate gut immunity. And certainly barrier integrity plays a role here as well. And I've included these lists in your resources as well, so no need to copy them down. From a dietary perspective, here are some basic guidelines that would be reasonable to adopt while treating gastrointestinal infections. Following a whole foods diet, including lots and lots of color. So we're looking for the polyphenols, myricetin, and gallic acid, as well as other constituents. Kale and berries for their quorum sensing inhibition, although that does fall under a whole foods diet, I thought I'd list it separately as well. Increased water intake, green tea for its many actions, fresh garlic, lots of culinary herbs and spices. So you've got cinnamon, cloves, ginger, rosemary, thyme, oregano, and black pepper, and others. And then decreasing sugar and simple carbohydrates. So choosing herbs, what do we do? Well, here's what I do. I look up the organism I want to kill and find out its dominant mechanisms of resistance. Then I tailor a botanical treatment plan based on that. So remember our 36-year-old female? We were going to use quadruple therapy to target her H. pylori because she had some resistance within her universal genes to amoxicillin and clarithromycin. So triple therapy was not going to be a good option for her. So we were choosing to use quadruple therapy. But we also wanted to address Klebsiella, which isn't classically susceptible to quad therapy. So a quick search of Klebsiella tells us that we could be addressing beta-lactamases, efflux pumps, biofilms, and membrane permeability. We could also be addressing histamine load, since many species of Klebsiella are potential histamine producers. Since we're not using a beta-lactam antibiotic, we don't need to be concerned about beta-lactamases, which leaves us with a few targets. We want to target efflux pumps, biofilms, and membrane permeability. So let's add in licorice to target the efflux pumps and biofilms, ginger, which again can target the efflux pumps and biofilms, Mahonia or Oregon grape has antimicrobial action, and Klebsiella is usually susceptible to the berberine-containing plants. It also has action against efflux pumps, biofilms, and can affect membrane permeability. And then let's throw in some green tea, just for good measure, because it does pretty much everything. Let's also add in a plant-forward, whole foods diet with lots of garlic, greens, and berries. To support terrain and normal flora, let's also do L-glutamine and probiotics in the evening. And this might not work for everybody. It might not be everybody's approach, but I like to use probiotics for any patient on antibiotics, both during and after therapy. One thing worth noting is that I would not be using tincture form for any of these herbs. And that's because since we've chosen quad therapy and we will be using metronidazole, any form of alcohol is strictly contraindicated. So you'll do better with water extracts or solid extracts of the herbs above, or just taking them fresh. So that's how I approach a case like this. It's pretty simple and can be very effective. And so here's the big take-home messages that I want you to leave with today. So test for resistance before giving antibiotics. Reserve antibiotics for serious infections. Tailor your treatments to the organism you're trying to kill. Use whole botanicals and use combinations of botanicals. And then again, support immunity and the terrain based on your patient's needs. Thank you again so much for joining us today for this webinar on antibiotic resistance. Your presence is so appreciated.